So if you were wondering how in the world are we going to get DNA out of feces, something we can actually test in the lab, we're going to be using a kit. It's from Zymo Research, and it has many, many steps, but none of them are difficult. You just have to pay attention and make sure you're doing things in the right order. So I'm going to break down what you're going to be doing very quickly, and then you'll have the protocol written out for you in the lab. So you're going to have a fecal sample that you add to these tubes that have something called bashing beads. The company's named them, in these collection tubes. And you'll add a lysis solution. Lysis just means to break apart. It's a common biology term. You're going to see it again. And you'll put that onto a vortex mixer, which is going to spin it around really, really fast. And then you'll centrifuge it to help pull all the material down back to the bottom of the tube. And so from there, you're going to pipette out, and I'll teach you how to use a micropipette. You'll pipette that into a collection tube that has an orange filter in the top of it. And you'll put that back in the centrifuge, and that, again, the forces in the centrifuge are going to pull that material down through the filter. And so you'll have liquid when you're done at the bottom of the filter, and you'll be able to pull the filter out and dispose of it. And to that liquid, you're going to add a DNA binding solution. And that will get put into another filter. This one's clear, plastic, and into another collection tube. And back into the centrifuge it goes. And what that should do is bind the DNA to the filter. And so the liquid that comes out at the end is not important. In this case, the DNA should be in the filter, so you discard the liquid, move the filter to a new tube, because you don't want to use that contaminated tube anymore, and put it back in the centrifuge after you've added a pre-wash buffer. You spin it again, and since we had a pre-wash buffer, you could probably guess there's going to be a wash buffer. You would add that, and again, spin that in the centrifuge. You're going to be getting some quality time with the centrifuge at this point. Remove the filter, put it into a new microcentrifuge tube, put it back into the centrifuge again, and what comes out at the end now is going to be pipetted through one last filter. This one's green in the kit we're using. You'll put that back in the centrifuge, and what comes out at this point, and it's a very good thing because I have run out of room, that is your purified DNA. And again, that's going to be DNA from the deer. It's going to be DNA from bacteria and viruses. So we're going to have to find the little fragment of DNA that we're interested in amidst all of those strands of DNA. And there should be a surprisingly large amount in that little bit of liquid that we're going to get out at the end of this process. So now we have our DNA, but it's a mixture of DNA from lots of different places, and so we have to target our gene of interest. And before I tell you about that, I want to just tell you a little bit about why DNA is amazing. First of all, DNA is a part of the central dogma of genetics, which says that DNA makes RNA makes protein. Proteins act like little machines in your body, and so you want them to always function the same way. And for them to do that, they have to be built from the same code each time. And so the code needs to stay the same. The code is also anti-parallel. So what that means is that there are little protein machines that work with the DNA, and they can only read it in one direction. Chemically, it's different. And so if you have a double-stranded DNA helix, and the little machines are working in both directions, then they're actually going to move in opposite directions. So if they're moving along both sides, they read the DNA in opposite directions. And so that fact that DNA is anti-parallel was used to create a technology called PCR, which is polymerase chain reaction. And we use that to copy fragments of DNA and make enough DNA copies that we can do things with them in the lab. And so if you have, again, that double helix and your targeted gene of interest, Then on either side of your gene of interest, let me back up, you can create primers, is what we call them, to stick to the double helix on either side, and then the little machines will come through and make a copy of the DNA in either direction. And so over time, we should get lots and lots of copies of that section that's in the middle. And so that's how we target our gene of interest. So now you have your gene of interest from your PCR, and so you need to visualize it. And we're going to use gel electrophoresis for that. Gel electrophoresis uses a thick gel that starts out as a liquid so you can mold wells into it. It's made of a starchy substance called agarose, and so it's a very firm gel, but it can still break. You have to be careful with it. And those wells don't go all the way through the gel, just almost to the bottom, so that you have this cavity that you can insert the DNA into, and it stays separate from all your other samples. The sample then, when you hook that gel up to an electric current, travels through the gel, and you can see it as a band. And the bands sort themselves out by length. So say you had 4 different lengths of fragments in a well, then it would separate to look like those 4 bands there. Or they could be 4 bands arranged in another way. But 4 fragment lengths, you'll see 4 bands. So in our case, the females are going to show 1 band because they have 1 gene length. The males should show 2 bands because the gene that we're targeting has 1 length when it's on the X chromosome and 1 length when it's on the Y chromosome. And so in the female, all they have is X chromosomes, and so they're going to have 1 band, but the male has an X and a Y, and so you'll see a band from the X that roughly matches the female's band, and a band from the Y that is shorter, and so it travels further through the gel. So that's what we're going to be looking at, and so when we finally get to this step, we'll be able to know if our fecal sample was from a male or a female deer. Pretty complicated, but hopefully every step's pretty simple. I know you can do it. And you're going to learn so many cool techniques that you can use in so many different studies. PCR and gel electrophoresis are kind of the foundation of a lot of what goes on in DNA science. So I hope you enjoyed learning about what we're going to be doing with deer poop this year.